Okay, so this is chapter 14, and I've decided to redo this lecture because I'm going to do it with the notes listed on the slides that we went through in lecture. I think it's a little bit more helpful for students. Okay, so broad spectrum antibiotic. The way the broad spectrum antibiotic works, it's in the name. It's a broad spectrum, so it will kill many different types of pathogens. However, it is very easy for a pathogen to develop resistance to it. And because of that, the ability for that pathogen to develop resistance to it, that can cause what's called a superinfection. Because as you see here, we've killed not only the surrounding microbes from this pathogen, but because we've killed those surrounding microbes, now there is no competition for that pathogenic bacteria. So then it's able to flourish in that particular environment. So that's what a superinfection is. So broad spectrum antibiotics can cause a superinfection by eliminating the competition with other microbes. So the best route of administration of an antibiotic, nobody should be shocked when I say it's IV here because you can generate the greatest concentration in the blood. But of course, we can't send a patient home with an IV in their arm and tell them to change their IV every 6 or 7 hours. So oral is what we typically give them in a pill, and you can see it's very difficult to generate high concentrations of the antibiotic in, or any drug for that matter, in the blood. So the social effects of antibiotic use. If you're Medicaid insured, you typically want to have your doctor prescribe to you antibiotics. Even if you don't know what it is infecting the child, you want your child to have antibiotics. And that's because people who have Medicaid typically do not understand, due to lack of education, that the antibiotics may not be the best solution and may actually make the situation worse by potentially allowing things like a superinfection to occur. Okay, beta-lactams. Inhibit cell wall formation without bacteria. Without a cell wall, the bacteria cannot resist osmotic changes and it kills the bacteria. So typically what we talk about is the penicillins. And the penicillins have this beta-lactam ring, which is this square structure with a nitrogen in it. So bacteria that are resistant to the beta-lactams make an enzyme called beta-lactamase. Beta-lactamase breaks that beta-lactam ring. When you break that beta-lactam ring, that allows you to render this chemical useless. So bacterial targets for antibacterial drugs. So typically what we see is like the beta-lactams, they target the cell wall. Something like vancomycin also targets the cell wall. Vancomycin is in particularly interesting because people who have MRSA, which is methicillin-resistant Staph aureus, you treat them with vancomycin. If they become resistant to the vancomycin, they get VRSA, which is vancomycin-resistant Staphylococcus aureus. Vancomycin is our last go-to resort antibiotic for MRSA, and if you get VRSA, then we have to look at, can we amputate that particular region that has the infection in it before it spreads to the rest of the body, leading to death typically. So this just gives you, I like this slide because it gives you an idea of where the drugs are targeting. So for instance, aminoglycosides, they target the 30S subunit of the ribosome of the bacteria. The sulfonamides, they target folic acid production. So it tells you where these drugs target. So when we start looking at things, so when I go back to the penicillins, for instance, you have to know whether they target gram-positive or gram-negative. Sometimes they target both, right? And the route of administration, you have to know. And when we talk about these drugs, mode of action, target, and drug class, inhibit cell wall biosynthesis, these are the beta-lactams. You must know these for the exam. So this table you must know. But I think these are pretty straightforward in that, for instance, aminoglycosides and the tetracyclines, these inhibit protein biosynthesis by targeting the 30S subunit. So fairly straightforward from things we've already learned in the course. You must know narrow spectrum versus specific. So that's narrow versus broad. So the narrow is specific. Know the pathogen to be able to prescribe a narrow. Broad, you don't have to know the pathogen, but you can have a person prone to superinfection. So this is bacterial cell wall synthesis inhibition. You must know the specific drug. You must know the spectrum of activity as well. This is continuation of that. Again, continuation of this. Inhibiting translation, so like we talked about before, tetracyclines, aminoglycosides, these are going to target 30S subunits. Chloramphenicol, macrolides, and lincosamides, these target the 50S subunit of the ribosome, but all of them inhibit protein translation. So you must know who's doing what to where. And this goes through it in the specific drugs that do it. You must know these. Again, I don't care if you know that's bactericidal or bacteriostatic. That's not going to be tested on. But you must know these, their mechanism of action, and broad or narrow. Sulfonides and trimethoprim, these do competitive inhibition, and so they're going to competitively inhibit enzymes that make nucleotides or nucleosides, which are going to be used as nucleotides. So normally you would have PABA, and PABA gets taken by an enzyme and converted into dihydrofolic acid, and that then gets taken by another enzyme and converted into tetrahydrofolic acid and then into a nucleoside, which becomes a nucleotide. So what the sulfonamides do is they competitively inhibit for the active site of that first enzyme, and because they bind there, then you can't make the second molecular structure, and then that second enzyme has nothing to work on. So it stops the chain from making nucleotides at the first enzyme. Now, if we look at trimethoprim, it stops the second enzyme from working by competing with this active site, so that when it binds to it, now it's no longer able to convert that into a nucleotide. So sulfonamide works at the first enzyme, competitive inhibition. Trimethoprim works at the second enzyme, competitive inhibition. And this just goes through it again and shows you the PABA first enzyme, trimethoprim second enzyme. And this shows you what a competitive inhibitor does, and this is like for the sulfonamides, so they bind directly to the active site. That way the substrate can no longer be bound to it. Antifungals, these are hard because fungus is a eukaryotic organism, so they're difficult to find. Antifungals that don't kill our cells. So because it's eukaryotic and we're eukaryotic, this is less different to us than bacteria, which are prokaryotic, and there's more differences between us and them, and so we can target those in antibiotics. We have cholesterol in our cells. Fungus have ergosterol, and so many of your antifungals will target that difference for ergosterol. So antifungal targets inhibit DNA and RNA synthesis, and this is fluticasone and